Hello everyone, a very good evening to all of you and welcome to the channel. So as the upper and lower PCS mains examination is going to be held in August, less than a month is left. So today we'll be covering a very important topic from the science portion, okay? There can be questions of differentiate between two terms that can be asked, and it has been asked in the previous years as well, so we need to prepare ourselves well for this topic. Okay, so we'll be discussing approximately 11 differentiate between questions today. Okay, if you want the PDF of the same, you can join the Telegram channel. The link is in the description and also in the channel description. Okay, you can join the link and you'll get PDFs out there. So let's begin. First question is differentiate between predation and parasitism. So predation is when the prey is there, okay, and there is a predator. Okay, so the predator is following the prey and eats it, so that is known as predation. Okay, act of preying by one animal on another. Whereas parasitism, it is a case wherein a parasite lives on the host's body. It can either live inside the host's body, known as endoparasite, or it can live outside the host body, called ectoparasite. Okay, so example is given: when the tiger is predating on a deer, it is known as predation, or of an act of you can give another example also. For example, a lion feeding on rabbit, or also herbivory can also be considered under this. Okay, whereas in case of parasitism, for example, tapeworm living inside the body of a human, or a lice living on the head of a human. Okay, then here what happens in case of predation is that the predator is larger and stronger, whereas in case of parasites, usually the parasites are smaller than the host. And in predation, no such metabolic dependency is there, but in case of parasitism, an intimate association is there and metabolic dependency, which means that for nutrition the parasite depends on the host. Okay, and finally, because of predation, the predator immediately kills the prey, whereas in case of parasitism, the parasite does not immediately kill its host. Okay, next question is difference between antiseptic and antibiotic. So antiseptics, they are effective against a wide range of organisms, whereas antibiotics, they are effective only against bacteria. Antiseptics, they inhibit the growth, but they do not kill the organism, whereas in case of antibiotics, they kill and thereby they inhibit the growth of the bacteria. Some examples of antiseptics are boric acid and iodine. And an example of antibiotic is penicillin or streptomycin. Antiseptics, they are usually applied topically, or they are applied outward on the skin. They are externally applied on the skin. Okay, whereas in case of antibiotics, they are usually taken orally in form of pills. Okay, they are usually taken inside the body. Next question is difference between plasma and blood. So blood is usually red colored, plasma, it is straw colored or yellow colored, and blood contains plasma plus the blood cells, that is the RBCs, WBCs and platelets, whereas the plasma lacks such blood corpuscles or blood cells. Okay, next question is difference between minerals and vitamins. Minerals, they are inorganic in nature, whereas vitamins, they are organic in nature. Minerals, depending on the need in the human body, can be classified into either macro minerals, which are required in higher or larger amounts, and micro minerals, which are required in lesser quantities. And vitamins can be differentiated, or they can be grouped under fat soluble vitamins or water soluble vitamins. Okay, an example of mineral can be iron or calcium, and for example, an example of vitamins can be vitamin A, vitamin B, etc. So what is the purpose of minerals? They can be needed as cofactors. For example, cobalt is needed for the formation of vitamin B, or they can also be directly involved. For example, calcium is required for the synthesis of bones, calcium as well as phosphorus. Whereas in case of vitamins, they are required for the proper functioning of the body. For example, for the proper functioning of the eyes, in order to prevent the drying up of eyes, vitamin A is required. Okay, next question is difference between rabi crops and kharif crops. So rabi crops, they are cultivated when the monsoon withdraws, that is when the monsoon is over by the month of October, then rabi crops are sown, and cultivation of kharif crops starts with the onset of monsoons. Okay, whereas and thereby rabi crops are also known as winter season crops, and kharif crops are also called the rainy season crops. The rabi crops are harvested in April or May, and the kharif crops are harvested in September to October. Examples of rabi crops are wheat, gram, mustard, and examples of kharif crops are rice, millets, maize, jute, and cotton. Next, difference between biomagnification and bioaccumulation. So biomagnification is the increase in the concentration of toxic chemicals at subsequent trophic levels. Let us take an example. Suppose there is a water body or a pond in which DDT has been added. Now when the fish, they take in the water, so DDT will start accumulating in their body. So that is known as bioaccumulation. Now when this fish is eaten by a larger fish, for example 5 to 6 smaller fish are eaten by the larger fish, and then the larger fish is eaten up by some bird. So the bird will have, now what happens is that in case of the small fish, suppose there was 1 mg of the DDT. Now there are 5 fish, so 5 mg of that DDT goes into the larger fish. Now each larger fish will have approximately 5 mg. Now if the bird is eating at least 2 fish, so it gets 10 mg of that DDT. Accumulation, not accumulation, the magnification has taken place. For accumulation in the smaller fish, only 1 mg was there, but the bird, when it is eating the larger birds, because of biomagnification, finally a lot of DDT has accumulated in its body. Okay, so that is known as biomagnification. So increase in the concentration at subsequent trophic levels is called biomagnification, whereas when it involves only one organism and only one trophic level, then it is called bioaccumulation. Okay, so we can also say that the difference is biomagnification involves several trophic levels and several organisms, whereas bioaccumulation involves only one organism and hence only one trophic level. Now the example has also been provided. Okay, for example, DDT concentration in fish and increased DDT concentration in birds when they are feeding on that fish, and bioaccumulation of mercury or even DDT in the smaller fish is known as bioaccumulation. Okay, involving only one organism and one trophic level. Next question is difference between plant cell and animal cell. This is a very common question. They can ask this question in the lower PCS mains examination. Okay, so the difference is that cell wall is present in case of plant cells, whereas cell wall is absent in case of animal cells. Then plastids are present in plant cells, plastids are absent in animal cell. Large vacuoles are present in a plant cell, smaller vacuoles are present in an animal cell. Then centrioles and centrosome are present in an animal cell, but they are absent in a plant cell. Okay, and carbohydrates, it is stored in the form of starch in case of plant cells, and it is stored in the form of glycogen in case of animal cells. Lysosomes are usually absent in case of plant cells and usually present in case of animal cells. Then the next question is regarding the difference between mixed cropping and intercropping. So what happens is that no particular planting pattern is followed in case of mixed cropping, whereas there are specific strips of crops. For example, the first crop, then the second strip will be of the second crop. So if a specific cropping pattern is followed, it comes under intercropping. First of all, in case of mixed cropping, the seeds are mixed together and then they are sowed without any specific row formation. Okay, and in case of intercropping, there is no mixing of seeds. So one row will be consisting only of crop 1 and the second row will consist of crop 2. Whereas in case of mixed cropping, they'll first mix the seeds and then they'll scatter the seeds randomly. So in one crop, there can be seeds of 2 different crops as well. Okay, and then in case of mixed cropping, the same fertilizers and pesticides are applied for all the crops, whereas in case of intercropping, different rows, there'll be a different application of pesticides and fertilizers. Then there will be a similar maturation duration in case of mixed cropping, so that all the crops or all the plants can be harvested together, whereas in case of intercropping, there is usually difference between the maturity periods. So what is the main function of mixed cropping? Why is it followed? It is done so that in case one crop fails, okay, for example, if a particular farmer has sown wheat along with mustard, and suppose there is some disease in the wheat plant, so at least he will be sure that okay, maybe the plants of wheat have been destroyed but mustard will still exist. Okay, so he can earn from the mustard crop. Okay, but the main purpose of intercropping is that it helps in the productivity of the crops. For example, if the farmer grows one leguminous crop along with wheat, okay, so the leguminous crop will help in nitrogen enrichment of the soil. So in that manner, the soil will be effectively utilized and still the nitrogen will remain in the soil because of the leguminous crop. So in a way, it will help in increasing the productivity of the wheat which is growing along with it. Okay, so it will help increase the productivity and thereby be beneficial economically for the farmer. Okay. Next question is difference between a prokaryotic cell and eukaryotic cell. So, in case of prokaryotes, as the name suggests, the nucleus is not well developed, whereas in case of eukaryotes, the nucleus is well developed. So, a well-defined nuclear membrane is absent in case of prokaryotes, whereas it is present in case of eukaryotes. Organelles are not well defined and not usually present in case of prokaryotes, whereas well-defined organelles are present in a eukaryotic cell. Genetic material is found in a region and that region is called nucleoid in case of prokaryotes, whereas genetic material lies inside the nucleus in case of eukaryotes. Prokaryotes, an example can be bacteria, whereas eukaryotic cell example is plant cell or animal cell. Next question, this has already been asked in the previous question of lower PCS mains. So what is the difference between DNA and RNA? Okay, so DNA stands for deoxyribonucleic acid, whereas RNA stands for ribonucleic acid. And the base pairs of nitrogen that are present in DNA are adenine, thymine, guanine, and cytosine, that is ATGC. Whereas in case of RNA, adenine, uracil, guanine, and cytosine are present. DNA consists of deoxyribose sugar, whereas RNA consists of a ribose sugar. Usually, DNA is double-stranded, whereas RNA is usually single-stranded. DNA is more stable, whereas RNA is less stable as compared to DNA. DNA, the main function in case of animal cells is it is the carrier of genetic information, whereas the purpose of RNA is usually that it is involved in protein synthesis, which is also known as translation. There are some other differences. For example, RNA, the three main types are tRNA, that is transfer RNA, mRNA, that is messenger RNA, and rRNA, that is ribosomal RNA. Whereas DNA, there are different kinds of conformations. For example, aDNA, bDNA, zDNA, etc. Okay. Next question is difference between artery and vein. This is also a previous year question. It has been asked in 2016 mains examination. So the arteries, they carry blood away from the heart, whereas veins, they carry blood towards the heart. Arteries usually carry oxygenated blood except for the pulmonary artery, and veins usually carry deoxygenated blood except the pulmonary vein. In the artery, the blood flows under pressure, which is very high, but in case of veins, the pressure with which the blood flows is very low. And valves are absent in case of an artery, whereas valves are present in case of a vein. Arteries are usually deep-seated, whereas veins are superficially placed. Arteries, they are bright colored or red colored because usually they carry oxygenated blood, whereas veins, they are blue colored or dull colored or dark colored as compared to the arteries because they are usually carrying deoxygenated blood. And the walls are elastic in case of artery because the blood is flowing under a lot of pressure, whereas the vessels and the walls are less elastic in case of veins. Okay. So these were the important differences that can be asked, or you can expect these questions in the upcoming mains examination. And stay tuned to the channel because we'll be posting more questions and more updates regarding Uttarakhand specific topics or some other topics which are or can be asked in the upcoming mains examination. So that's it for today, and always remember, whatever exam you're preparing for, you can do it. So that's it for today. We'll meet in the next video. Till then, bye-bye and take care.